talks which hopefully will outline some interesting new data on efficacy of the VI conjugate vaccine in the model, some data on the immunology and some of the underlying biology related to human infection with Salmonella typhi. I think it promises to be a very exciting session, ending with back to NTS with Cal, so it should be a very good afternoon. And I thought I would just start by introducing again the challenge model, which I can't do because the slides aren't working. Can we put the first talk up?
And then right this forward.
Thank you. So just to introduce the challenge model. Now the challenge model that we use is actually a fairly ancient model in that it was first used 50 years ago at the University of Maryland, and you can see Ted Woodward there, who was coordinated those studies. And the Salmonella strain that has been used consistently through these studies came from the lady there, Mrs. Quayles, and is known as the Quayles strain. It was used for most of the challenges done in Maryland in the 60s and 70s, and has been used in all of the Oxford typhoid studies that we're going to talk about. There is one person in the audience who was involved in those studies, and that's Mike Levine, and he very kindly helped us back in 2008-9 to start the work to set up a challenge model in Oxford. And so our model is very similar to the original one that was started 50 years ago, but with a few differences. The first is that the strain is manufactured to GMP, and here you can see someone making it to GMP, then transferred to our facility, frozen in vials. And on the day of challenge, we make up the Salmonella strain in a bicarbonate buffer. First of all, the volunteers drink some bicarbonate to neutralize gastric acid, which gives a much smoother take in the challenge model if gastric acid is neutralized. And then after that's happened, they then drink the Salmonella typhi, and we follow them for the next 2 weeks to see who develops typhoid. And in the model, about 2/3 of people who are challenged will develop Salmonella typhi. So the primary endpoint that we'll be presenting in the studies today is a fever greater than 38 degrees, plus or minus a positive blood culture. And we do take blood cultures every day to try and identify anyone who is developing typhoid. So some people don't have any fever, some have a fever. And I just want to highlight that that's quite different from the situation in the field, where the definition of typhoid is fever for 3 days and then a positive blood culture in that individual. So it's quite a different setup from typhoid in the real world. And of course, we assume that we're giving quite a high dose because 2/3 of people develop typhoid who are exposed. So there's some differences from the field. Now, just to very rapidly rehearse the disease phenotypes, and this is revision for all of you because Tom showed these slides yesterday. Some individuals don't get typhoid, and the way these tables are set up is the black line is the fever, or at least the temperature here, no fever. The green line, for those of you who can see it at the bottom, is the C-reactive protein. And then there's an orange blob there, which is stool culture. And in people who are develop bacteremia, there will be red squares as well. And here's an individual who's developed typhoid. You can see a rise in fever in the C-reactive protein. And this individual has positive blood cultures at the time of the fever, and they also start shedding in the stool, as you can see here with the orange squares. This is an individual who didn't develop fever, but did become bacteremic. And so this is someone who's developed typhoid, but is asymptomatic. And we've had some Oxford University students who've done their final exams whilst looking rather like this. They don't do any better by having typhoid. This is an individual who has typhoid. They've developed fever during the challenge period and a rise in their C-reactive protein, but no positive blood cultures. So there's various different disease phenotypes that we might see in the model. And this is an interesting group who have persistent shedding without any evidence of developing typhoid. So they've developed their persistent shedding some time after the original onset or the challenge, but didn't develop any symptoms or positive blood cultures. So in our program that we've been now running since the first challenge in 2011, we've challenged a total of 370 people, and there's a series of different studies which are listed on this slide. We've done some vaccine studies. So first of all, there was a dose escalation study. We undertook a vaccine study, and we presented some of the data from that yesterday with 2 live attenuated vaccines. Selena in a moment will talk to you about the typhoid conjugate vaccine study, but just to highlight there that we're not going to present today, but also use the model to test different antibiotics. So it isn't just for looking at vaccines. We also have a paratyphoid model which has been set up. Malik is going to talk a little bit about re-challenge with typhoid and paratyphoid, and there's a new program looking at a strain where the toxin has been knocked out, which is currently underway. So I've been asked by the organisers to remind you that on your desk you have surveys which have to be completed. And it's very important that with this very international group that we have representing Oxford, you give them high scores, because at the border now, if you don't get a high score at a conference, they don't let you back into the UK.
It's that bad.
Yeah, it's that bad. Okay, so Marcelo, take it away with cellular responses to the challenge model.
Okay, I would like to start by thank you, Andy, and the rest of the group for inviting me to share some of the data that we generated at the CVD using cells obtained in this Oxford model. So that saves me the time to actually go through the model, which is very useful. But what I decided, because of the time restrictions of this presentation, is to focus on message as opposed to on data. So we can discuss data as much as you want later on, but for the time being, I will just focus on these general guidelines that I think we can learn from this challenge model. And what I show in this slide is that, of course, when we do vaccines, we try to enhance immunogenicity and decrease reactogenicity. But more and more and more, what responses are we measuring? That is becoming a key issue. So it's clear from some talks yesterday and some talks today that in reality, we do not know what are the correlates of protection in typhoid. Antibodies are probably important because they probably will have an activity when typhoid is extracellular, but CMI will be critical to actually eliminate typhoid from the host. So over the years, mostly using orally immunized typhoid vaccines, candidate typhoid vaccines, and TY21A, my team has demonstrated the presence of CTL activity and by many techniques, chromium release, granzyme, CD107 expression, interferon production, and that these responses were mediated by CD8, but also CD4 have the capability of inducing killing. Now, the CTL activity, in addition, was restricted not only by the classical 1A molecules, but also by non-classical HLA molecules. We also went on to show that they are really type 1 cytokine responses, that is the pro-inflammatory type. The cells do home to the mucosa and to non-mucosal tissues based on homing markers and flow cytometry. And also, we described the presence of long-term multifunctional HLA-restricted CD8-positive cells that are able to produce cytokines. So we demonstrated all of this over 2 decades or so, but the question remains, which of these CMI responses, if any, are actually associated with protection from typhoid fever? And this is very important more than from a theoretical point of view, because if we can define which are the correlates of protection, mechanistic or non-mechanistic, what we can do is zero in into which responses we want to emphasize as we move into the development of new and better vaccines. Also, they can help predict long-term protection, and we can accelerate the development of broad-spectrum vaccines or bivalent or even trivalent vaccines. So it came a point 2 or 3 years ago that we we went into a collaboration with Andy and his team at Oxford, because with the re-established challenge model, we finally had the opportunity to try to identify which immune responses actually are correlated with protection. And I emphasize correlated as opposed to mediate, because in humans, unfortunately or fortunately, we have to deal with associations. So the only thing I will mention in this human challenge model slide is that I focus our efforts in trying to identify the correlates of protection by studying cell-mediated immunity, and this is the T effector, the T regulatory cells, for which no data existed, T memory cells, mate cells, antigen-presenting cells, both macrophages and B memory cells, and to study, if possible, long-term immunity after the volunteers were treated with antibiotics. So what makes this unique is that some volunteers developed typhoid and some volunteers did not develop typhoid. So the approach was to try to compare the responses on those volunteers who developed typhoid and those volunteers who did not develop typhoid, trying to get closer to identification of which are the correlates of protection. So just very few data slides. Memory and effector T cells. When we look at baseline, this was the first surprise, and this is what I think that we should focus on, is that we saw that at baseline, what was the status of the responsive CD8 positive responses? They are the ones that, in a way, were associated and differentiate who developed typhoid and who did not develop typhoid. And as you can see in this slide, you see CD107, a marker of cytotoxicity, showing significant increased responses. Same thing for TNF-alpha, IL-2, MIP-1 beta, and even strong trends for interferon gamma and IL-17A, which are obviously critical responses, as we just heard, in the gut mucosa. But this is also very, very interesting and enlightening. If you look at the TD, that's the blue lines on the left and the right, for interferon gamma and TNF, what you see is that after challenge, there is a marked decrease in the cells that are responsive in the periphery, probably because they are homing to the gut or to extraintestinal site, which we demonstrated by also having integrin alpha-4 beta-7 in the panel. But what is even more interesting is that the no typhoid disease volunteers, they were flat. It was not variation in the rate in circulation. So the conclusions, well, obviously we have much more data that I couldn't show, but also we demonstrate a delayed time to diagnosis in the subjects who developed typhoid disease as the responses were higher at baseline, and also that what really correlated with response is very discrete sets of multifunctional cells. Now, we also were very interested in studying regulatory T cells, because I am a firm believer that what in the end will result in protection is a balance between regulatory T cells and effector T cells. Because if you have too much effector cells, you might have reactogenicity as opposed to protection. So just one slide of this whole set of data, and this is remarkable because at baseline, if you follow the blue line with the asterisk, the volunteers who developed typhoid disease had very high levels at baseline of integrin alpha-4 beta-7 S-typhi responsive cells. That means that those cells had the capability to home to the gut. If you have downregulatory cells homing to the gut, that provides a putative explanation of why these volunteers develop typhoid. In contrast, the no typhoid people, they have increased regulatory cells over time. So these are the conclusions. This also was the first demonstration that T regulatory cells are capable of functionally suppressing S-typhi specific responses. Very important because there were no evidence of Tregs having an effect in this system. And among the other things that we study are antigen-presenting cells, monocytes, dendritic cells, and the like. So if we look at the level of activation of monocytes, as measured by CD38 positive expression and CD40 expression, You can see on the left panels on the volunteers that developed typhoid disease, there was an extremely high degree of activation at the time of typhoid diagnosis and up to 48 and 96 hours later. This is in circulation. However, in the monocytes from the no-TD volunteers, these are the two right panels, you can see that it's absolutely no movement. It's like they have not seen the bacteria. If we look at alpha-4 beta-7 expression, we see exactly the same thing. In the volunteers who developed TD, there was an enhancement of the expression of this molecule that tells the cells to go to the gut. However, the no-TD volunteers have no effect. But not always the no-TD volunteers are flat. I am showing just one example in which monocyte at day 1, and this is the right panel that is circled, you can see a strong increase in the percentage or in the intensity of the binding of a fluorochrome labeled Salmonella typhi in the volunteers who developed no TD, but only after 1 day. And this was absent in the volunteers who developed TD. Of course, there is an increase in the binding on the volunteers who developed TD at the time of typhoid diagnosis. That points to another constitutive function of the host in controlling Salmonella infection. Much more data is presented in a series of papers that we published in the past couple of years. And I will stop here just to mention that there is a mate cell T-cell paper in press because mate cells are very, very important for mucosal immunity. And what we have observed in following these close kinetics of the expression of mate cells and mate cells activation, that in reality, those mate cells also become very activated, but only in the volunteers who develop typhoid. Like in many other responses, the no-TD volunteers just they don't seem to see the bug. So just to summarize it, because I have to fulfill my promise to be in 12 minutes, and I have only 1:35 to go. What I believe is very important that we are balancing the immune response, that is the effector T-cell responses and the regulatory T-cell responses. What happens if we have too much regulatory responses and too little effector responses? Probably ineffective protection. What happens on the contrary? We might have reactogenicity. But of course, nothing is so simple. We might need and might want to approach homeostasis by defining how the new attenuated typhoid vaccines are generated. But as you can see, the effector responses are not only CD4, CD8 responses, because each of them have many, many subsets, some of them extremely active, either pro-inflammatory or suppressive. And the same thing is true for regulatory T-cell responses. You have the classical Tregs that are FOXP3 positive, CD25 high, but there are many other new subsets that are very active. And then, of course, we have the humoral immunity, which for sure plays a role. Antigen-presenting cells, I show some data on that end. And of course, we cannot forget the host HLA factors, other genetic factors, nutrition, and a factor that might be extremely important, which is the microbiome. And we are doing an extensive series of studies on the microbiome, and we will show the data that is coming close as soon as possible. So this is the overall conclusion. Identifying the correlate of protection is critical. We need to dissect what is happening between the non-protective or downregulatory immune responsive from the ones that are actually effective. Because if Salmonella typhi is one thing, it's a stealth organism. It loves to hide from the immune system inside macrophages and many other cells. And that is critical because those mechanisms of protection from the point of view of the bug are very difficult to overcome by the host. And I would like to thank the many, many people that are involved in this project. From the data I presented today, Stephanie Fresnay, a postdoc in the lab, worked on the T effector and memory responses. Monica MacArthur, an assistant professor, worked on T regulatory cells. Franklin Toapanta, an assistant professor, worked on B cells and APC. And then the rest of the people in the lab involved in this project, and of course, Andy Pollard and the Oxford team. Thank you very much for your attention. And it was 12 on the dot.
Pardon?
It was 12 minutes on the dot.
Very good. Thanks very much, Marcelo. Any questions for Marcelo? I mean, you showed the association with CD8 cells and a suggestion at baseline that was associated with protection. Mm-hmm. How does that fit with Lorena's data in the mucosa? Because you were suggesting they were migrating to the mucosa, but when they get there, they don't seem to be protecting, at least the ones she's looking at.
Well, I really will need to look at the data in more detail than a quick presentation, but I think that's perfectly reasonable. I mean, the fact that there are increases is very reasonable.
So the increase fits in with the finding.
Yes,
the
because
homing.
they are disappearing from the blood and going to the mucosa, and I think that the key will not be as much as a whole subset of cells. The key will be the granularity of the response. And what are those Tregs doing? Are they going to the mucosa? That will be a critical question. I don't know, are you looking for T regulatory cells in the mucosa?
I did look for them. I tried to phenotype them, but there are not that many, at least in the first part of the intestine, in the duodenum, there are not that many Tregs, so I cannot do a deeper analysis.
Which technique have you used?
Sorry?
Which technique have you used?
It was just by flow cytometry. I was looking for them.
Maybe we have to talk a bit later.
But I guess there's also a timing issue that the biopsies are at not the same time points that you're looking at, so that there may be...
yeah, obviously the biopsies cannot be done that close to the diagnosis time. Has to be weeks before and has to be at least 3 weeks after the challenge.
The mucosa is an extremely active site that with regulatory and counter-regulatory signals all the time. So I am not surprised that they will switch on and off within a very small period of time. Yes.
So Cal.
Marcelo, thanks. Lovely summary. Obviously, the pre-existing immunity seems to be very important. I was intrigued by your monocyte binding that you showed at the end. Did you look for association with that and with pre-existing antibody as well? And as a whole, did you sort of link in what was happening with the humoral immunity with your T-cell responses?
Well, that is a question that probably Andy should answer, because they already published that the antibody responses, anti-LPS or anti-H, do not correlate with the disease outcome.
But specifically with your monocyte binding that you were showing there?
Ask me the question again.
So you showed a peak in monocyte
Yes.
binding amongst those who, early on amongst those who didn't develop typhoid fever, and I just wondered if that was independent of what was happening with antibody or not.
I don't know.
I think that Marcelo showed at the time of diagnosis there was a rise in those who developed typhoid. Everyone was challenged. This wasn't at baseline. This was at the time that they developed typhoid.
Okay.
On the TD volunteers. On the no TD, there was that peak at day 1.
Okay.
So I'm Rezwan again. I have the questions to the whole panel. I have a question to the whole panel. Sometimes while working with the typhoid, we know that Salmonella typhi has a great ability to suppress the immunity, and that could be one of the ways it evades the immune system. And that's why sometimes it could explain that why natural infections doesn't give much of the immunity like others. Are you looking into that part of the typhoid fever from your challenge studies, that how it evades the immune system?
I can just give a quick announcement on that. Anna Lucino, who is sat there in the audience, she has a poster on transcriptomics on dendritic cells that we have infected with 2 different strains of Salmonella typhimurium, LT2 and D23580, and we compare them with the cells that were in the same well, but they are not infected, and then we compare them with the transcriptomics of uninfected cells. So although it's not perhaps that related to the model, we are looking into assess suppression and so on by the infection.
Okay, thanks very much, Marcelo. So we're now going to move from typhoidal Salmonella to their cousin, and Cal is going to talk about that.
Thank you, Andy, Tom and Malik, for giving me the chance to talk about something which I have no data on, but we're through the first round, the preliminary round of a grant application with the Wellcome Trust, and I'd very much appreciate the input of this audience as we move through to a full application. So just in terms of overall aims of what we'd be trying to achieve with a NTS challenge model, we'd essentially like to establish something similar to what Andy and the team have established already for Typhi and Paratyphi A, for two main broad reasons: to understand pathogenesis and immunobiology of NTS infections in man better, and also to accelerate the development of NTS vaccines, particularly those that you've heard about already, bearing in mind that there might be some spin-offs for diagnostics as well. So there's been some talk sort of behind the scenes for a while about the possibility of having an NTS-controlled human infection model. Do we need one? Well, I think the disease burden that's been around for a while and is continuing to grow suggests the importance of an NTS vaccine for Africa. And the problem we have at the moment is that our knowledge of human NTS infections, particularly invasive NTS infections, are quite limited. And in terms of vaccinology, just thinking of the presentation that Malik gave in terms of background knowledge, we're 100 years behind typhoid vaccines, given that the wholesale killed vaccines for typhoid were given back at the beginning of the 20th century. So this has been touched on already, modalities of protective immunity, and the same questions arise for NTS as they do for typhoid, except with the absence of human data from challenge models and from vaccine studies. So just to point out again, these are facultative intracellular bacteria. They've evolved to survive within macrophages, but capable of cell-free survival. And so this is consistent with the role of T cells to clear disease, as Piero was talking about elegantly earlier from the mouse model, and also antibody to, particularly to prevent fatal bacteremia. So what knowledge we do have at the moment for NTS infections, a lot of this comes from mouse studies, and NTS has been a great model for mouse immunologists. We also have quite a lot of data from patients with primary immunodeficiencies, and a limited amount of data so far from cases and at-risk populations in low and middle-income countries where the disease is prevalent. So there have been a number of reviews on this. I was privileged to be involved with a review with Piero early on in my time working in this field, looking at similarities of data and information from mouse models and individuals with primary immunodeficiencies. That's moved on somewhat. We had a review of the immunodeficiencies and genetic susceptibility a couple of years ago. But one thing that's striking is that the clinical presentation in humans with primary immunodeficiencies is often quite different from that of what we see with individuals with INTS disease in Africa. And then just in terms of what we see in mouse not always being applicable to what we see in man, we've been working on bactericidal antibody a lot, and we're able to basically find out that and show that what we could see in humans with bactericidal responses wasn't the same, wasn't mirrored in mice. So there are subtle differences, although we've learned a huge amount from mouse models. So moving to populations where NTS is a problem, and from work that we and others have been doing in Malawi and other countries, there's been evidence that's been pointed out already in terms of the immunoepidemiology to infer an important role for antibody, both bactericidal and opsonic. Although work that we did with humans with HIV infection in Africa suggested that perhaps you might have too much of a good thing, and very high levels of antibody against O antigen of LPS were associated with blocking. And then, of course, we've now got 2 vaccines in development, and great presentations in the previous session on both of those. But it would be great to have a better understanding of protective immunity as we move forward with those. And one of the questions is antibody enough? So the vaccines are very much geared towards producing antibody responses, particularly to O antigen, also to flagellin. But it's important to point out that both of those vaccines contain protein components of Salmonella, the flagellin with the MacLennan vaccine, the whole outer membrane protein array with the GEMRA approach, which potentially gives a range of antibody responses and T-cell help that we wouldn't get if we had say a conjugate with a carrier protein which wasn't Salmonella specific. So animal models indicate that T-cell immunity is required for full elimination, and perhaps studies from HIV infections support that as well. And then we've also got just this issue of inhibitory antibodies as well. So a lot of questions that perhaps a model of NTS could provide some accelerated answers to, and to at least increase our knowledge and understanding. And there's certainly a good opportunity at Oxford with all the hard work that's gone on establishing the model for typhoid and paratyphoid infection models. And there's also just a general increase, as I'm sure many of you are aware, in the development of controlled human infection models and how to best approach how best to get maximum amount of data in terms of immune responses, etc. So I think one of the big questions that we've had for a long time, and it's come up several times during this conference, is what is it about S-typhimurium ST313? What's the relative contribution of that pathovar to the invasive disease in relation to host immunity, stroke immunocompromise, and transmission that we still know relatively little about? And a controlled human infection model would allow us to examine that without the interferences of these other factors, impaired host immunity and transmission, which are obviously very integral to what's happening in the field. There are challenges with this whole question, with this whole idea of setting up a model for NTS. INTS disease is, by definition, invasive. But what we'd be obviously needing to do would to give oral administration of Salmonella. Now, as John Crump pointed out in the plenary sessions at the start, the original portal of entry is almost certainly through the GI tract, but we don't know so much what's happening after that. Is it staying there in the ST19s in developed countries? Is it going through with ST313 infections to become invasive? What we do know from studies in recent years and work by Robert Ansari, Sam Kariuki, and what Satish presented earlier on, is that these ST313 isolates are found in diarrheal infections as well as invasive infections. Safety, I think, is a growing concern, even though typhimurium is classified as BSL2 and typhi as BSL3. I think just things like the level of mortality that's seen in the field with these invasive infections does require some attention. So, are we at risk of putting individuals in danger by having an NTS challenge? One thing that's quite striking, certainly in adults that we look at in Africa, is we very rarely see bacteremia in immunocompetent adults. Children, it's a little bit different, and you're dealing with immunonaivety there, but you're not going to have young children in challenge studies. We don't know about a latent established disease state, but work that Pierre has been doing, work that David Holden have been doing, and others suggest that there might be a persistent state going on. And just again, from the clinical observations by Melita and Chineri and others, again, focusing very much on those with HIV, this is another question that's unresolved at the moment. So in terms of sort of safety and trying to mitigate that, we'd certainly want to keep a very close eye on volunteers and look at them perhaps 12-hourly as opposed to 24-hourly as been done with typhoids, and have obviously used strains which are sensitive to ciprofloxacin, ceftriaxone, where we can use prompt antibiotics and have those readily available. And I think the other question is, what would the endpoints be? So I think we'd certainly want to borrow what the typhoid and the paratyphoid challenge models have already, so the fever parameter greater than 38 for 12 hours, or if we had a positive blood culture for Salmonella typhimurium. We just don't know at the moment whether there are these subclinical blood cultures occurring. We certainly know if we sort of do increasing frequency of blood cultures, we occasionally pick up positive blood cultures without symptoms, which we didn't think we were getting before. And I think we're going to have a gastroenteric endpoint as well, and there needs to be some question about quite what we use for that. Would it be 3 or more loose stools in 24 hours, the classical diarrhea description, or something else? So in terms of the high-level outline of what we'd be aiming to do, without going into the details, which are very much paralleling what's been done already with the other models, we'd need GMP manufacture of a Salmonella typhimurium. And I think this is one of the biggest questions: which typhimurium would we do? Ideally, if time wasn't an issue, perhaps we'd go to ST19 and see what happens in a common and garden strain from the UK before then going to ST313. But time's an issue, funding's an issue, and these models aren't necessarily cheap. Then we'd want to go on an established infecting dose that would give a sort of 65-70% infection of volunteers, so with a dose escalation. And then obviously sort of use all the tools that have been developed already to characterize the presentation, pathogenesis and immune responses, and ideally from this get a better idea of correlates of protection, susceptibility, in a way that we just haven't been able to do so far for the NTS infections. And perhaps as well get clues in terms of diagnostics, which, as for typhoid, we ideally need for NTS as well. And then I think finally, to finish off, what would the next steps be? Well, obviously, having established a model, we'd love to be able to use that to look at efficacy of candidate NTS vaccines. And then finally, just one thing that's sort of becoming more topical at the moment, and that's come into the recently announced Wellcome Trust strategy on human infection models, is potentially transferring the model to a developing world setting, potentially to Kenya, potentially to Malawi, to then see what would actually happen in individuals in the endemic context as well. And obviously safety being a very important issue to establish first of all. So I'll finish there, but I just want to acknowledge my gratitude to Andy and all of his team for the discussions that they've given me already in terms of getting this so far, and the discussions with others in this room and elsewhere, such as Gordon Dugan and Mike Levine. So I'll finish there. Thank you.
Thanks very much, Cal. So we have time for a few questions. Melita.
Hi. Cal, you're talking about immunocompetent individuals here, is that correct?
Yes.
How do you get around the problem that what you're essentially going to be setting up is a model of enterocolitis, and the higher the dose you push, what you will produce is more and more severe inflammatory mucosal disease and profuse diarrhea, with perhaps some overspill into blood, but an incredibly different pathogenesis compared to what we see in the disease that you're trying to prevent, where there is frequently no diarrhea at all and very prolonged bloodstream infection. I'm not sure how translatable across, and I'm not sure how far you will be able to push the model with the degree of mucosal disease that you're likely to generate in this.
Well, I think the two key words there are not sure. So I think at the moment we just don't know, and I think this has been one of the arguments against going to look at this so far. I think what we need to do is to basically look. So at the moment we haven't been able to really study. There's no challenge model existing so far to study this, and there's a balance here between safety and invasiveness. Obviously, if we could be completely sure of our safety, then we'd go ahead and we'd do this on HIV-infected individuals. Now, I don't think anyone would want to go there, at least not to start with. So I think it's going to be very much a learning agenda in the first instance, and that's why I think it would be very welcome to have input of other people who've got thoughts on this. So, I think we need to try it. And then I think the other thing is just, is there a difference between ST19, ST313 in terms of what will happen?
Okay, thanks, Carlos. We've got 2 more questions, and it'd be great if you could say not sure for both of those, so to save some time. Mike.
Yeah. Needless to say, I would love to see a ST3113 model set up, particularly for evaluation vaccines. So I'm totally supportive of the concept. But I'd like to share a little bit of, can't say gray hair because I don't have that much left, but a long experience of setting up a number of different challenge models for the first time in humans. So this one is with a bug, where our experience in Mali, in that special set of kids, but not HIV infected, we have great respect for these bugs, and clinically, we don't understand them, really don't understand them. Even the doublins, the INTS Salmonella doublins that were completely antibiotic sensitive, there's a high case fatality. So with all the unknowns, my suggestion is that you approach this in a stepwise fashion. And rather than go right in with a wild type ST313, which is short incubation, and I would say, I don't care about gastroenteritis with ST313. I want to know about how it gets into the blood and whether you can treat that quickly and that it doesn't go any further. Towards that end, I would suggest that you have ways to do this stepwise and to build up confidence on everyone, because a lot of people are going to be affected. So by the results, some of the things you can do, there are ST313 attenuated strains already, multiple attenuations, and that have been tested in rhesus macaques. One thing you can do is evaluate those. Second thing is to take an ST313 and make a single mutation. Many years ago, Doug and I collaborated, taking a Typhoid strain with his cosmid clones, and we made the deletions and gave a vaccine strain that did not cause illness, but when you went up in the dose response, it caused vaccinemia. And that was a clear objective readout. I think having that as a readout as an intermediate step would be extremely helpful, because you could then test parenteral vaccines against preventing vaccinemia. So I am very supportive, but I would say with a couple of steps in between, and time and money shouldn't be an impediment if you know, there are questions. If I'm the only one who's raised trying to, you know, go stepwise, then I'm in a minority, but I say this from a lot of experience.
Thanks, Mike.
Thanks very much
for the discussion earlier.
Thanks very much for the comment. We won't have a response to that, but I think that's very helpful. Laura.
So, Cal, I just want to take Mike's ideas a bit
And
further.
very short.
And before going into doing a vaccine study, the community really needs to understand what you would be looking at in this model to avoid any false negative, false positive elimination of a vaccine, since you don't have any positive control to say that the model is appropriately characterized. So
Thanks.
really it's the qualification of the model itself that needs a lot of attention.
It's a learning agenda to start with. Thanks.
Melita, you've already had one go, so as long as it's only 3 words long, you can ask a question.
Okay, we know from Malawi 313 actually causes plenty of plain old diarrheal disease without being invasive, and it's also carried asymptomatically in adults and children. So there is the possibility that the main difference between 19 and 313 is going to be in the invasive phase, and they'll both cause diarrhea and you may not see much difference. I'm not sure how you get around that. But the idea that it's intrinsically invasive, particularly in an immunocompetent person, may not show much difference.
Thank you.
Thanks very much, Cal. So I think this has been a great session. I think we've seen some really interesting new results and lots of slides with blue and red and black spots on them. And so I'd like to thank Selina, Malik, Giorgio, Lorena, Christoph, Jenny, Marcelo and Cal for fantastic talks.